Good evening, everyone. Welcome to the Craddock Gardens Science Cafe. My name is John Evans. I'm the Conservation Horticulture Manager here at APG. And it is truly my great pleasure to introduce my former Thesis Committee Chair, Dr. James Hill Craddock, who will be speaking to us tonight on the conservation of the American chestnut in the southeastern United States. And just to let you know, there will be a short question and answer session after his talk tonight. And I would like to remind you also to please turn off your ringers on your cell phones so that we can go through it smoothly. And a little bit more about Dr. Craddock. Dr. Craddock is the UC Foundation Davenport Professor of Biology at the University of Tennessee at Chattanooga in the Department of Biology, Geology, and Environmental Sciences. He grew up in Woods Hole, Massachusetts on Cape Cod. He's the son of a marine biologist father and an emergency room nurse mother. And he went to sea for the first time at age 16, working summers on commercial fishing boats, which is something I just found out about tonight. And he described it to me, and it was pretty much what you would expect. He grew his first chestnut tree from seed he planted at age 15. And obviously he's still quite enthusiastic about chestnuts. He moved to Italy in 1987, where he and his wife Paola helped run their family's restaurant business. They moved with their son Emilio to Tennessee in November of 1994. Dr. Craddock completed doctoral and postdoctoral research on hazelnut and chestnut biology at the Università di Torino in Turin, Italy, before conducting postdoctoral research on anthracnose-resistant dogwood cultivars at Tennessee State University. He holds a master's degree in horticulture from Oregon State University and a bachelor's degree in fine arts and biology, double major, from Indiana University. So without further ado, we'll bring on Dr. J. Hill Craddock.
Thank you, John. Can everybody, is it okay if I talk this loud? Can you hear me? Then I won't have to use the microphone. So thank you for the introduction, John. Wonderful. He really was one of my graduate students, and we got to reminisce a little bit about that. It's really, really, you can imagine what a pleasure it is to see former students, you know, making the world a better place, which is how I feel about. Come here. Should have skipped this part. Anyway, the work I feel, the kind of work that we're doing is conservation. I think we're making the world a better place. So keep up the good work, John. I should have skipped that part. Okay, so conservation. You know, you might have heard. I'm gonna tell the story of the American chestnut tree a little bit. I didn't know where to begin this story, so I'm gonna go way back, so like to deep time. We talk about restoration. You've maybe read about the American chestnut. And you know, John and I talked about this a little bit at dinner tonight. What does restoration mean? And I'm very skeptical of the term because I don't think we're going to restore the American chestnut to what it once was. That's gone. That's in the past. So that's why I want to frame the work we're doing in conservation. In other words, try to protect what we have for a possible future. But let's not pretend that we're going to put the world back to some previous state. That I don't think is a realistic goal. And so we might save ourselves from that effort. You know, we a lot of effort about arguing about what restoration means if we just don't use that term. So I've been working for many years now, almost 30 years, with the American Chestnut Foundation. And we have as our mission restoration of the American chestnut. In other words, we're trying to put the American chestnut back into the forest where it ought to be. And so I just want to reframe the conversation a little bit from restoration to conservation. So when I say back deep time, I want to take a little bit of a global view. The American chestnut is just one of many species of chestnut. There are some number, I don't know, 7, 8, maybe 10, 12 species. I brought examples of 2 here you can look at. I brought some show and tell. By the way, somebody mentioned auction. The starting bid on this one is $1,000. If anybody wants one, I'm kidding. That's a joke. This one is a species that's native to the southeastern U.S. This is a chinkapin. They grow naturally as a shrub. That one's from actually Florida. Grows as a tiny shrub. That's about as tall as they get in nature. The American chestnut, of course, was a giant tree, a forest giant. The reason I've got pictures of the fossil record up here is because in the literature, there's some evidence that the center of origin, in other words, the center of diversity of the genus Castanea, in fact, maybe the whole Fagaceae family, was ancient East Asia. And there's some evidence to support that sort of westward migration of the genus across evolutionary time, and then the speciation into North America from the Far East, but maybe not. In fact, some of the oldest fossils we have that look like chestnuts come from North America. In fact, maybe some of the oldest fossils of chestnut itself are North American, not East Asian. It may be, of course, that the fossil record is incomplete. We just don't know about the East Asian stuff. But it's certainly plausible that the rocks in this part of the world harbor, that may be the part of the continental plate where chestnut first arose from an evolutionary point of view. And I mention this because this is deep time. This is tens of millions of years ago. So we see a long evolutionary history of chestnut in North America. Of course, it was present in historic times. Here's a set of maps that my co-author Taylor Perkins, who's here, and I prepared for a book chapter we wrote recently. I don't know if it's projecting very well, but I've got European, 1 species, East Asia, some 4 species, depending on the author. There may be more than that. And then North America, I've got 3 species here listed. Dentata, that's the American chestnut. Castanea pumila, the Ozark chinkapin. And Castanea pumila, variety pumila, the regular old chinkapin. Speaking of chinkapins for just a sec, though, and I'll talk more about chinkapins in a minute because I'm a little bit crazy about chinkapins. Had a grad student a few years ago who looked for historical names that have been applied to this species. And I'm using the term species in a very broad sense because the chinkapins are highly variable. They vary from tall trees in the Ozarks to shrubs in the Florida, peninsular Florida. She found 23 different taxon names. In other words, 23 different names have been applied to that plant that we call chinkapins. Now, I don't think there's 23 species, but I think the story is more interesting. So here is a set of slides just to illustrate what some of the species are. The trailing chinkapin in Florida, that's this one. And I brought this one to show you because it's sort of one of the extremes. It's the smallest of all the species. What I call a regular old chinkapin, the Allegheny chinkapin, that one happens to be from Georgia. That's a photograph from Georgia. A very broad range. If I go back, you can see the range of chinkapin is in blue on this map. This is Texas. This is Louisiana, Florida, all the way up into Maryland, and maybe even as far north as Long Island. There's possibly as far north as New York, Staten Island, New York, maybe. The Ozark chinkapins are tree size, nearly extirpated from chestnut blight and land use changes. Very, very rare, limited to the Ozark uplands of Missouri, northwestern Arkansas, and extreme eastern Oklahoma. Very interesting problem. We'll talk about that later. The American chestnut, of course, is the iconic American chestnut that everybody knows about. And here's what I'm going to talk a little bit more about today, which is the Alabama chinkapin, which is a species that we rediscovered. It was first named in the 1920s by a botanist from North Carolina. He changed his mind a few times about what it really was. Ideas about the Alabama chinkapin have changed back and forth over the last half a century or so, and we think we have good evidence to support it as a thing. It's a thing. Whether it's a species depends a little bit on how you define species, but I'll talk more about that in just a minute. Because we're talking about conservation, we're plant breeders. So I use the term germplasm, meaning that that's the stuff from which I can make a new plant. So as a plant breeder, it could be pollen. Pollen is germplasm. It could be the plant itself. You could take cuttings. If it's a sexually reproducing plant, of course, you can pollinate it. You can get the seeds and grow the seeds. So germplasm is a broad term. That's the term that we use to refer to the stuff. And in this case, it's the living collection. All right, so that's the living collection. So I've divided this table into naturally occurring and introduced species. And notice that Castanea dentata, which is the American chestnut, occurs twice on here. It's a naturally occurring species in the eastern North America, but it's been introduced into the West. And so if you go, in fact, the national champion American chestnut is in a suburb of Portland, Oregon. Okay, they're introduced in the West. They're natives, but they're not native to Oregon in that sense. They were taken by people who moved westward with them and took their trees with them. So I've got other species on here. I've got Craddock, which is a Japanese chestnut, sativa, a European chestnut, Mollissima, which is a Chinese chestnut, Henryi, Seguinii, some other species from China you might have seen on the map that I showed earlier. And these are important because they represent a lot of effort. In other words, it took effort to get them here. Now, plant introductions are fraught. We all know about the risk of exotic invasive pest plants, and you've got weeds in your garden. I'm sure that you use very endearing terms with them in your own garden, right? But this represents a human effort to bring diversity to North America. And so I think when we talk about conservation of Castanea resources, as a plant breeder, I'm interested in these other species too, the introduced species also, because it's in those species where the genes for disease resistance may lie. And so the great tragedy of the American chestnut is it was destroyed by a biological invader, a fungus. That was a biological invasion and accidentally introduced pest organism. But the Asian species have the genes for genetic resistance to that disease. And so we can breed chestnuts for disease resistance, taking advantage of the natural resistance of Mollissima and Craddock, for instance, and Henryi. Seguinii is a shrub. We're not going to use that probably, but Henryi, a tall tree, Mollissima, a common tree. So this is the talk I'm not going to give tonight. I'm just showing this slide to remind me to tell you not to talk about this. So we're not talking about chestnuts as a nut crop. Although I love chestnuts, I love to eat chestnuts, and historically, traditionally, chestnuts have been an important food source for humans in traditional cultures in East Asia, in Europe, and in the Americas, in North America, going back to the origins of humanity. We know from archaeological evidence in East Asia and China and Japan and in Europe that our ancient ancestors were using chestnuts. In fact, in parts of southern Europe, in Italy and Spain, Portugal, and the islands of Corsica, there was a veritable chestnut civilization. Okay, so chestnuts are delicious, they're nutritious, they can be stored like a dried grain. In fact, it's the grain that grows on trees, and so a tremendous economy, a tremendous wealth developed around. This is the talk I'm not giving. Okay, and just to remind you, this is the second slide in the talk I'm not giving. You'll have to invite me back if you want to talk about chestnuts as a nut crop. We've done some research in Chattanooga and Tennessee about chestnuts as a nut crop. This is a really good one, by the way, Payne. If you wanted to grow chestnuts and make money, I think you could grow this one. They're really good to eat. That's a Chinese chestnut. That's Castanea mollissima. They're resistant to the chestnut blight. They're resistant to the Phytophthora root rot disease. So the American chestnut, of all the species, Castanea dentata is the one largest tree. It grew taller than any of the other species. They're great long-lived trees. We don't really know because most of what we know is anecdotal or has been reconstructed through some careful archaeological work. very, very abundant, but don't believe everything you read about the American chestnut. I'll say that again. It's a story that gets better with every telling. You know, it's this like, they get bigger every time the story gets told. They get more numerous and like 1 out of every 4 trees. No, that's just not true. Okay, there was a paper published a few years ago by a group at the Harvard Forest about pre-blight abundance, where they did some meta-analyses of historical records, and they were debunking a lot of this myth, the American chestnut myth. And I put that on the American Chestnut Foundation listserv email, and I got a note from one of the vice presidents that said I was hereby immediately excommunicated for challenging the dogma. So really, chestnuts are important, and they were important historically, mostly because of their abundance. It's true that there were places in the mountains in eastern Tennessee, western North Carolina, northern Georgia, where the chestnut canopy was probably quite extensive. In other words, it was the dominant tree in that ecosystem. They had a broad range from Mississippi all the way to southern Canada and coastal Maine, mostly an upland tree. They were a lumberman's dream. The tree on the left was cut for a timber volume study. It was actually cut in 1911. That's from Connecticut. And if you know about lumber, the whole thing went to the sawmill, right? And that tree was 110 feet tall and was 80 years old. 80 years old, 110 feet tall. The one on the right is a picture from western North Carolina. That's from Mitchell County, North Carolina. And there's a human in this figure. There's a person standing here. You can't really see him, so I've enlarged him a little bit to show you. So it was in the western Carolinas, northern Georgia, southwestern Virginia, where chestnut reached its greatest abundance and its greatest size. So that's where it was the largest and most abundant, were in the mountains of the southern Appalachians. All of that changed in 1904 when a new disease was discovered. The disease was discovered in New York City, in the Bronx. Plant pathologists from Brooklyn went all the way over to the Bronx to look at the trees. They were in the zoo at the Bronx Zoological Garden. At that time, they were large, old American chestnuts that had been planted, were dying of a disease. He isolated the fungus. It's an orange mold. He described it as a new species. The species name has changed a few times, but now we call it Cryphonectria parasitica. That will be on the quiz. So learn that one, Cryphonectria parasitica. It's an ascomycete fungus. It kills the bark. It eats the cambium of the tree. These orange pustules are one of the fruiting bodies are in there. That's called stroma. And you can see where I cut there with my knife. The necrotic tissue actually extends in front of the mycelium. The fungus kills the tree tissue before it eats it. So it's great. First it kills you, then it eats you. Which means it's not a very sophisticated parasite. It's actually misnamed in that sense. It's a necrotrophic fungus, and it can live quite happily as a saprobe on dead bark for many years. In fact, that's probably one of the ways it spreads around the world. Okay? In fact, we're pretty sure that the fungus got to Italy on chestnut logs from blight-killed trees that somebody cut and then shipped off to Italy. The Italians were building railroads, and we know there were 3 points of introduction of chestnut blight in Italy: Naples, the port of Naples; Trieste, the port of Trieste in the northeast; and Genova, in Genova. And then the chestnut blight started in Italy, doing what it did in North America. It's hard for us to imagine the scale and the scope of this destruction. We're talking about billions of trees, over thousands of square miles, millions of acres of land. It was a true pandemic in the sense that every individual was affected. There were no unaffected individuals. It completely removed the American chestnut from its ecological niche as a forest canopy dominant. Okay, they're literally wiped them out. We can talk more about that. By the way, we'll talk more about that later. There's another disease. It's called the Phytophthora root rot disease. It's caused by an oomycete, was a fungus-like microorganism that lives in the soil. The Phytophthora, by the way, that's hard to say. Yeah, Phytophthora. Not everybody can say that word. It has a PHTH in the middle. It's worse than chestnut blight because it also kills the root system. The American chestnut tree is not killed by the blight itself, which only attacks the trunk. If the roots are growing in an otherwise fertile site, they can resprout from the base. In fact, they're legendary in their ability to resprout and grow from a cut stump or a damaged stump. The same with chestnut blight. The root rot disease is really bad. It's also known as ink disease. It's been present in the southeast since the mid-19th century, probably introduced on crops from East Asia, from South Asia into Georgia and South Carolina, North Carolina. Spread through the Piedmont and the coastal plain, so that even by the late 19th century, chestnut had already been removed from the Piedmont. Of course, there was agriculture and land clearing for agriculture, but this disease is really bad. American chestnut in particular is very susceptible. This is the talk I'm going to give tonight. So that part of, that was the story of the chestnut blight real quick. Here's what we're doing. Conservation of rare Castanea genetic resources depends on. We have to find the trees. They're out there. We have a network of volunteers and enthusiastic people that love American chestnut for one reason or another. Their grandfather told them about it, or they've learned about it recently, or they're naturalists and they're using some phone app or something, and they're in the woods, they see a tree. So we have a tremendous network of information out there to find the trees. They're not extinct. There are millions of American chestnuts out there. Occasionally we see large surviving ones. This is an LSA, that means a large surviving American. And we'll talk more about what that means in a minute. We have to find the trees. I want to characterize that diversity. And what I mean by that is I want to know about it. I want to know how it is that it's, what it is. Is it an American chestnut? Is it a hybrid? Is it some new thing? We're using molecular biology tools to help us do that. We're studying the genetic diversity using DNA sequencing technologies, very powerful population genetics computational tools to allow us to look at the population as it physically exists on the landscape, the structure of the diversity itself to find areas of high diversity. If I've only got a few conservation dollars to spend, I want to focus those conservation dollars on populations that are more worthy. In other words, if I can target areas for collection into areas where there's more genetic diversity, I get more conservation for my dollar. Does that make sense? Right? Plant propagation, we can grow the seeds if we can get them. They don't root from cuttings. They're really, really hard to root. In fact, pretty much impossible to root from cuttings. But we can graft them. So I'm going to talk a little bit about grafting. And of course, we can make hybrids. And so the hybrids allow me to capture some of the naturally occurring diversity in the form of a blight-resistant hybrid. Of course, it's not an American chestnut anymore. It's a hybrid American chestnut. Okay, but by clever plant breeding, I can dilute out those exotic genes. If I select for blight resistance in every generation, we can recover at the genomic level so that the trees are mostly American. In other words, we can recover a lot of the American, the genetic diversity that naturally occurs here, we can conserve in the form of hybrids. Let me put it that way. It's not on the list here, but there's also a genetic engineering solution. I'm going to talk a little bit about the genetic engineering solution because we frequently get questions about that. Not everybody thinks it's a good idea, but it has tremendous promise, and so I'm just going to introduce that idea. This would be a GMO chestnut, a chestnut tree that's been genetically engineered to be resistant to chestnut blight. So we'll talk about that. Looking for chestnuts, we have a lot of information at our fingertips. Literally, you've got a phone in your pocket that has lots of information on it, right? I can look for old herbarium records, historical records, witness tree lists, actual documented locations from these amateur phone ID apps, lots of things. We can use geographic information systems to help us predict where chestnuts are. They like acidic soils. They like certain aspect, and sort of a northeast facing. They like a certain elevation. They like a certain rock substrate. We can predict where we're going to find them, but then you got to walk in the woods. And so we look for them like the FBI looks for a dead body. We walk shoulder to shoulder. Literally, when we find them, we map them. We can take a tissue sample. The tree on the left here this is the American chestnut tree. That picture's old, by the way. That's about a 25 years old photo. They're all grown up now. Those are my students, right? They've all grown up now. A colleague of mine, Joe Shibig, sampled sprouts on the Cumberland Plateau in Tennessee and counted the growth rings. A stem 1 centimeter in diameter had 35 growth rings. 35 years old to get about that big. This one's probably not that old. We know from the biology of the chestnut tree that that plant was probably alive when chestnut blight came through, because the chestnut tree only blooms in full sun. They don't bloom in the forest understory. It's too shady. They've been suppressed as suppressed saplings. That tree was probably a sapling when chestnut blight came through in the 30s, and it's still a sapling. Occasionally we see something like this. Christine and Walter here, other former students. Christine is pointing, I like this picture because you can see that the stems are coming up in a semicircle. You can imagine that that's from the crown of a dead tree. Parentheses. Probably, we know this from some work done by Fred Paillet and his colleagues, ecology, chestnut ecology, that the large trees that were killed by blight toppled over. And when they toppled over, they pulled their roots out of the ground, they died. So most of the surviving trees out there in the population were probably saplings or seedlings when blight hit. But here, anyway, you can imagine that this is where there was a big trunk, and these are the trees that have come up from the, just use your imagination. Pretend that's what that is. This tree didn't die. It has blight, but it hasn't died of blight. So there's a couple things going on here. A similar phenomenon was first observed in, by the way, here's what we call them, large surviving Americans. This one is in Lincoln County. By the way, if you hear me use the term beautiful canker, you know I'm talking to a plant pathologist, right? Plant pathologist, oh, beautiful canker. That's a great canker you've got on there. So this is a big ugly canker. It's not killing the tree. The tree is surviving. So a couple things are happening here. It was first observed in Italy, this phenomenon of trees that had blight but didn't die. Italian biologists thought that the trees had somehow acquired immunity. It was a French guy, Jean Grant, who hypothesized instead that the fungus got sick. A case of the enemy of my enemy is my friend, right? So at the time, this was in the 1960s, late 50s, 1960s, no one had ever seen a fungal virus. Maybe they had some in some yeasts, but in filamentous fungi, there were no known examples. Now we know that all organisms have viruses. Fungal viruses are common now. But back 50 years ago, that was, nobody knew about that. So he hypothesized, he called it transmissible hypovirulence. And so here's the virus-free strain. Here's the same strain grown with two different viruses, the CHV Euro 7 virus and the CHV EP713 virus. The fungus is debilitated by the virus to the point where it cannot kill the tree. So the virus keeps the fungus from killing the tree. So the tree can live. And so back to this slide, we isolated bark. We collected bark. We isolated the fungus. You know, a student in my lab did this. We sent the isolates off to West Virginia University where they were studying the hypoviruses. And it turns out it's a double-stranded RNA virus. It's a naked virus. There's no protein coat, and it's transmitted by fungal anastomoses. In other words, from fungal cell to fungal cell. There's no airborne particle. You can't catch it. In fact, it's a little bit tricky to transmit. And it works very well as a biocontrol in Europe. In fact, on your next trip to Italy or France or Spain, you might see whole mountainsides covered with chestnut forests. And those forests are there in large part due to the presence of these hypoviruses. It hasn't worked so well in North America. In other words, in the United States, we have not seen the landscape scale change that we saw in Europe. So it doesn't work so well here. But we do find these large surviving Americans. Here's an example of some large surviving Americans. This was an experiment done by Gary Griffin at Virginia Tech back in the 1970s. He had some of these large survivors. He took cuttings from them and grafted them into a woodland, and then he inoculated them with the CHV, the CHV1 Euro 7 virus, and They're still alive today. That's a picture I took just the other day. Remember 2017? That was just the other day. This tree has blight. That's a canker. It's got blight from the ground all the way up into the canopy, but it's resisting the blight. Why hasn't it cured blight in North America like it did in Europe? It could be that the American chestnuts are more susceptible than the European tree, and so they just die too fast. And what Gary told me was that Gary thinks that American chestnuts are susceptible to blight, but they're not uniformly susceptible. And that there might be enough variation in susceptibility that there are trees that can express this hypovirulent trait. In fact, not all trees will do this. If you just go out and collect wild type Americans, it's difficult to keep them alive. So it may be these large survivors may be special. Okay, so here's the work we've got going on at Chattanooga, which I think is why you invited me to talk, to hear what we're doing. So we've got a nursery. We're plant breeders. We make a lot of crosses. We collect as many seeds as we can. The BNF2 thing is another lecture. That's about chestnut breeding. I'm going to talk about that later. We're trying to grow seed orchards from which we can produce the seed that could be given to landowners, mostly on an experimental basis, to plant chestnut trees, to reintroduce them to the forest. We're doing what I'm calling the small stem assay, which is we're trying to shorten the generation time, trying to shorten trying to speed things up using a small stem assay. I'm going to mention that again too. And of course we have new genomic tools that we didn't have available to us until very recently to literally map the genes for resistance. 30 years ago when I started this project in earnest, we, the chestnut breeding community, we made some assumptions. One of the assumptions was that it was feasible because there would be few genes involved. If it was 1 or 2 or even 3 genes, we could select for blight resistance in a few generations. We could fix that resistance. In other words, we could get homozygous resistance, and then the progeny from those would all be resistant. Would that it were so simple. It turns out it's a lot more complicated. In other words, there are many genes that control blight resistance in the Chinese chestnut. And we've got some evidence of some contributing genes from the American side. So it's not as simple. In fact, it's much more complex than we thought it was 30 years ago. But we still are making progress. We've learned a lot about chestnut breeding. And I'm going to show you some of that work. We're producing as many as we can of the American chestnuts for what we call germplasm conservation orchards. And those will be important in the future, particularly if we solve the problem in some other way. Parentheses, for example, the genetic engineering solution. If genetic engineering becomes a viable option, we can use these germplasm conservation orchards to be the mother trees for the GMO pollen. We might be able to do that. We're grafting trees. I'm going to show you some pictures of grafting, long-term phenotyping, breeding and selection for Phytophthora root rot resistance. And then I'm going to talk about the chinkapin work. In fact, I think I'm going to talk about chinkapins first. I took this photo at the trailhead of the Pinoti Trail at Cheehaw Mountain State Park on the southbound. They've got this beautiful arch, and they've got all the different species of trees. They've got about 25. These are ceramic plates, ceramic tiles that has the name Castanea pumila, and they've called it the Allegheny chinkapin. I'm going to have to write to the artist and tell him you messed up. That's an Alabama chinkapin. We know it's an Alabama chinkapin. It looks like an Alabama chinkapin. We can recognize them, and I'm going to talk about that more in just a minute. So Castanea pumila is not the American chestnut. It's another sister species. Occurs broadly from Texas and the coastal plain all the way up into the Ozark Plateau, and then eastwards all the way into Maryland and then down to peninsular Florida. I showed you the map. Several years ago, I had a grad student who was looking at chloroplast genomics, did a study on, so the chloroplasts, by the way, chloroplasts, which are the green part of the plant cells, they have their own DNA. kind of cool thing. It's a really small piece of DNA. It's a little circular piece of DNA. Looks like a bacterial piece of DNA, but it's very, very abundant. So it's kind of easy to study. In other words, in a leaf cell, there's lots of chloroplasts. You get a lot of DNA out, so it's kind of easy. And she was able to do some work on the genetic diversity of the chloroplasts. One of the things that she found was that the chloroplasts, different from the way we expected, the chloroplasts were shared among species. And we didn't expect that because chloroplasts are inherited maternally. So each species is supposed to have its own chloroplast. And we thought we were going to be able to use the chloroplast to identify the species. In other words, it has a chinkapin chloroplast, it's a chinkapin, has a chestnut chloroplast. It didn't work that way. And we found out that the situation was very complicated. Well, at least we were confused. In some places, the top photo there, we saw chinkapins and American chestnuts growing side by side, literally one on top of the other, where they were morphologically distinct and easy to tell apart. Okay, and we call that sympatry. They were growing together in sympatry. In other places, we would find one or the other species, but there were some populations, and here geographically very close together. That's northern Georgia, that's Floyd County, Georgia. That's Cherokee County, North Carolina. Relatively very close geographically, something very different. We didn't know what it was. And so there are a couple of hypotheses. One, it's a chinkapin and we're ignorant. We just don't know what chinkapins look like. That's a valid hypothesis. Two, it was some kind of hybrid. We were not the first ones to see this plant. The hybrid hypothesis had been proposed 100 years ago. Someone said it looks like a hybrid between American chestnut and chinkapin. It's got to be a hybrid. Other people said they were Japanese chestnuts. Somebody else said it's an Ozark chinkapin, from out west. Taylor, I'm just going to point him out over there. Wave, Taylor. Taylor said, I've seen these before in the herbarium. W.W. Ash described this species as Castanea alabamensis in 1925. Thank you. The details like that I depend on Taylor for. So 1925, W.W. Ash, he changed his mind a couple times. By the 1980s, this extremely diverse in terms of morphology group of plants we call chinkapins had all been lumped into one species. Castanea pumila, and then two botanical varieties were recognized. Variety ozarkensis from the Ozark uplands, which is really distinctive, and variety pumila, which is the regular old chinkapin. There was an author, George Johnson, who was actually a student in North Carolina, but was from Arkansas. I think he was from Arkansas. Thought that these Alabama chinkapins were actually Ozark chinkapins, which in that way he kind of agreed with W.W. Ash, but they'd been extirpated. In other words, by 1988, Johnson looked for them, he couldn't find them, he declared them extinct in Alabama. Well, we found them. We had them in our own collection, we didn't know they were there. Then we went out to look for them. We can identify them. Now, I'm sorry I didn't bring one. I had some in pots, but they just weren't ready to show you. The bottom of the leaf, the lower leaf surface is glaucous, which means it has this whitish covering on it, which is actually a waxy cover. It's not hairy. So the chinkapins, in fact, when you come up and look at this one, you'll say, oh, these also are whitish on the bottom. I think you can see that. But these are whitish on the bottom because they're hairy. They've got tiny hairs. They've got stellate trichomes. These are hairless. Now, they do have hairs, but the hairs are on the veins and on the margin of the leaf. They've got this ciliate margin. So we can recognize these things in the field when we see them. We've been able to map their locations. We're trying to answer the question, are they a hybrid? So we used, nowadays we have more powerful tools. Taylor had at his disposal much more powerful tools than Megan Binkley had. We went back in the field, we collected again. So here are some of our sampling sites. We sampled very heavily in the southern Appalachians. These red dots are the areas where we sampled alabamensis. And I told you before, they're a thing. So here's a phylogenetic tree that shows relationship. And by the way, this is to scale. The length of the nodes, the distance between nodes is the relative genetic distance. And you can see American chestnuts form a group down here. We call this a clade. Those are the American chestnuts. The chinkapins are all up there. Unfortunately for this paper, we only had a few specimens of Ozark chinkapin. They're there. Allegheny chinkapins and alabamensis. So you can see they're definitely a thing. They're chinkapins, but they're not regular old chinkapins. And then these other are outgroups. The stars are hybrids. Another way we can do a genetic analysis is to look for introgression, which is the mixture of different species. And this is a statistical program that tells you how many kinds there are. It's sort of arbitrary. Here we've labeled them by species because we knew by looking at them what species they were. We can tell dentata apart from, here's a dentata type. We can tell them apart by looking at them. Here's a genetic analysis that shows us, according to this analysis, there are two types. There's the chinkapins and the chestnuts. But notice that the alabamensis show no evidence of dentata. Interestingly, some of the pumulas do have some evidence of dentata in them. So there may be some hybridization between dentata and pumula. Or that variation there, that mixture that seems like it's a hybrid, could be a vestige. It could be vestigial. It could be a relic of some ancient diversity. When before pumula and dentata were different species, we're going back tens of millions of years, there may have been many genes that were mixed up. And so in population biology, we call this incomplete lineage sorting. In other words, they just haven't been all the way sorted out yet. So we've got a current project we're working on to try and figure that out. To do that, we needed more samples from Arkansas, so I went back to Arkansas. I went in 2019, I went again in 2021. The guy in the middle here is Logan Estes, a PhD student from the University of Arkansas, wrote his thesis. The title of his thesis was, "Where are the chinkapins?" I'm not kidding, that was actually the title of his thesis. Where are the chinkapins? So he was very handy to have in the field because we just followed him to the chinkapins. It was a wild trip. I also had the opportunity to travel with Fred Paillet. Here's Fred's most recent book, Ozark Forest Forensics. When you walk in the woods with Fred, he's like, "Don't move that twig." It's a crime scene, right? He's reconstructing. By the way, by training, he's a paleoclimatologist. He studies paleoclimate. What use is it to study paleoclimate? If you know about paleoclimate, you might be able to predict what's coming, right? So Fred's interest is in chestnut, was because chestnut pollen is a very good indicator in the paleo record of a temperate, humid climate, because chestnut trees have this actually kind of a narrow, they grow here, but they don't grow everywhere. And so if you see chestnut pollen in the fossil record, you know you had a temperate woodland environment, right? So anyway, it was quite moving. This is the boneyard of Ozark chinkapin. They were wiped out by chestnut blight, but because it's so dry, it's a very high elevation, it's not Cumberland Plateau, Ozark Plateau, the dead trees are just lying there. It was quite striking. We did find survivors. We collected from 3 areas. We collected in northwest Arkansas, southwestern Missouri. By the way, these are not all the collection sites. These are the ones for which we have the DNA extracted and the sequences ready. And each one of the squares actually covers up multiple collection locations. And we collected 5 plants per location because we want to do within population diversity also. Then in the south here, in the southern Arkansas, we were looking for alligators. We did not see an alligator. This is regular old chinkapin. These are not Ozark chinkapins. They're different. And so this area here in the Ouachita Mountains, we think may be a transition zone. So we were very interested in collecting across this whole area. Meanwhile, back east, here we are in the southern Appalachians. This is Cumberland Plateau, and then these are actually outliers of the Blue Ridge. We've got the populations of Alabama chinkapin, and then in the Chattanooga area in Tennessee, so far, we have not found Alabama chinkapin in Tennessee, but we're looking. So we're still trying to answer this question about gene flow between the species. And so the statistical test we're going to do, and I love this one, it's called an ABBA-BABA test. Don't ask me questions about the ABBA-BABA test because it's over my head. Taylor knows about it though. He'll stick around afterwards. You can ask him about the ABBA-BABA test. But what it basically does is it's a test of gene flow between these groups. And so we can pool our DNAs. Remember, this is all molecular biology, so it's DNA sequences. How many DNA sequences do we have, Taylor?
Human genome.
No, in this one.
About 500 whole genome sequences, 500 plants.
500 plants. Okay, how many SNP loci are there?
About 30 billion.
30 million SNP loci. So the point of me saying that was just, you're going to go, wow. Also, you can't do it on your laptop. Right? You need to plug it into the mainframe, like we're talking Oak Ridge or something like that, right? And so it's kind of tricky to do the thing. So some other interesting chinkapins. This is the actual one I brought. I'm fascinated by these things. I want to go back. We've got the start of an interesting common garden study here. These ecotypes are very unusual. These grow in a fire climax. They get burned frequently. One of the questions is, are they small because they get burned, or are they just small and they happen to live in a place that gets burned? But you can see this is our local guide. That's Bob Simons, a naturalist from Florida, is pointing out they grow in large genets, so they propagate themselves clonally by sort of spreading underground. He's got one genet on one side is about 2 feet tall. The other one on the other side is about 3 feet tall. Fascinating biology. Large areas in these sand hills in central Florida were covered by this chinkapin. By the way, I've been to Florida a couple times. Everything down there was trying to hurt you. It's like it's got spines or thorns, or it's venomous, or right. It was this horrible place to work, and it was really hot. But I'm going to go back. What?
Or the governor.
We're not supposed to talk about that today. I'm changing subjects. I didn't have a nice gradual transition to this one. So this slide reminds me I'm going to talk about something else, which is that Phytophthora root rot disease that really kills the chestnuts. And so they don't sprout back from the base when the roots are dead. So when they get Phytophthora, they're gone. Phytophthora can persist in the soils for decades, maybe centuries. We don't know. It has a very broad host range. It attacks lots of plants, unrelated plants. When it's in the soil, it basically means you cannot grow chestnut trees there, American chestnuts. So one of the tests, if it's an agricultural soil or a former agriculture soil, and I want to know if there's Phytophthora out there, we use the American chestnut tree bioassay, which means I give you a chestnut tree, you plant it in your field, it dies, you've got Phytophthora. We can pull it up, you can see the roots are gone. It eats the roots off. Also, pine voles will eat the roots off too, but that's a little bit different symptom. We can tell if Phytophthora has killed it, and the American chestnut is so sensitive, that's actually a really good assay. In the laboratory, it's a fastidious fungus. It's not really a fungus, it's a microorganism. It's difficult to grow in culture. It needs a very rich medium. So to get it out of the soil, we use a rhododendron leaf bait. In other words, you incubate the soil with a piece of rhododendron leaf, and then you grow the fungus out of the rhododendron leaf. It's kind of a trick. Taylor and another student of mine isolated, this is a Phytophthora symptomatic orchard. All the trees had died of Phytophthora. It's very heartbreaking because that was decades of work. I mean, literally 10 years of my life, and a landowner who had donated their land and built fences and fertilized and watered and mowed. All the trees died of Phytophthora, which means the experiment we were doing was on blight resistance. We got no data. In other words, you can't even answer the question, right? We planted these trees to find out if they had blight resistance. They all died of Phytophthora, so no. For me, because I'm a teacher, it's curriculum. I love this picture because I use my students. It's actually coercive technique. If you're going to get a grade, you have to do the work, right? We inoculated the trees as a class project. It's a pretty easy thing. If the roots are all dead, that's a no. The ones that have lesions on the roots, these two pictures, they're probably going to die anyway, but some of them are really resistant. And so we planted the resistant ones back into the orchard, and that was this photo. This is actually, it's a winter, but you can see vigorous new growth. We planted some 400 trees that had been all the way through the killing tubs. These are grown in tubs in big tanks, the killing chambers. Most of them died, but the ones that survived look really good, so we're breeding for Phytophthora resistance. I've got two outcomes from that work. One is that we've got the resistant progeny in the field growing that we can now evaluate for blight resistance in the future, but we also identified the parent tree from which that tree came. In other words, the resistant ones, not all the progeny we tested were resistant. We were able to identify the mother trees that transmitted that resistance. And by the way, we've done this in Virginia and North Carolina, South Carolina and Georgia and Tennessee. So we've got, this is just part of a much larger program. So here, this is a tree this is Cochrane 7-30. As a tree that we know progeny from that tree had had Phytophthora root rot resistance. So I can use that tree in my breeding program. So here I am, here's an American type. This is actually a blight resistant tree, American type. I've got a colleague here, that's Clint Neal in the top of the bucket, who's pollinating using pollen from my Phytophthora resistant tree to incorporate blight resistance and Phytophthora resistance together. By the way, if you know about probability and statistics, we're doomed. If you're betting on this, you're going to lose money for sure. In fact, selecting for two diseases at the same time is really daunting. It's really daunting. And the numbers are staggering, and that's why we have to grow literally thousands of trees to get some good ones. So, switching topics again, this is a tool we use for conservation, grafting. So why we graft, I'm not going to read you the list. You can read it yourself. But you know that grafting is a method of cloning. I can go out into the woods, or better, I can send my grad students out into the wilderness. They bring me the branches back. We can graft them in the nursery. I can get all of that genetic diversity in one row in the nursery that would be sometimes hundreds or thousands of miles apart. If you consider, from Alabama to Virginia is hundreds and hundreds of miles if you have to make multiple trips in a vehicle. For instance, you have to go out in the summer to identify the species. You have to go back in the winter to collect the cyanwood. And so grafting is a method of cloning. I use a seedling rootstock, which we can grow, they're a dime a dozen. We use different kinds of grafting, but essentially I'm splicing together the top, which is the scion, onto a rootstock, so I have a dual organism. The advantage, of course, is then we can do hundreds of these in a greenhouse environment. We can grow them in containers in the nursery, so here they are in a high light environment. They can be fertilized and watered. I can treat them if I have to with fungicide or insecticide. There's a close-up of the graft union. When they bloom, I can collect, in this case, these are male catkins producing pollen. It just takes a few catkins. In other words, from a few flowers, I can make hundreds of seeds. And so I'm able to capture then, like I was saying, I can capture this genetic diversity. The tree here in question, Alabama, Jefferson County, this comes from the Talladega National Forest. This is truly in the wilderness. It was Trent Deason, the wonderful guy, hiked, he's still alive, he survived. Had to hike way up in the mountains to get this cyanwood and bring it back to me. We took the pollen to the Meadowview Research Farms in Virginia, where they had some American chestnuts that were blooming, and we made American by American cross, so we can grow these in a conservation orchard. We can also grow them we could also use that same pollen on a hybrid if we want to capture hybrids. So some of the breeding work we do is in the wilderness. Here's a colleague of mine, Sam McInturf. We're in the Slick Rock, Joyce Kilmer wilderness area, which is the Tennessee-North Carolina border. This is true wilderness. There are no man-made structures in that view up there. This was an area that had been hit very heavily a few years prior by a pine bark beetle. And so most of the pine trees had died off, and that released the American chestnuts. They grew up very quickly into the light gap. Many of them bloomed. They all bloomed together. There were some large survivors up here. By the way, I didn't find these trees myself. I've got local informants. Ross in Alberta took me to these trees, and I asked Ross, I said, when did you see these trees? And he said, Alberta, when was that when we saw these trees? And she said, that would have been about 1964. Ross in Alberta, they're in their 90s. He walks like a tortoise, but he goes the same speed, uphill, downhill, level, it doesn't matter. Amazing character, took us right to those trees. There is a GMO approach. This is the genetic engineering part. And so this was worked out by a group in New York at the State University of New York in Syracuse at the Environmental Science and Forestry Laboratory. They took a gene from the wheat plant. It's the oxalate oxidase gene. And it's ubiquitous, which means it's in lots of plants. It's not truly ubiquitous because it's not in chestnut. Chestnut doesn't have this enzyme, but lots of plants do. And it detoxifies oxalic acid. So oxalic acid is one of the chemicals that the fungus, Cryphonectria parasitica, uses to kill the tissues of the American chestnut. It may actually be that the American chestnut is responding to oxalate by killing itself as a sort of misguided defense, because if it was a normal parasite, many parasites need living tissues. Cryphonectria doesn't. Anyway, it's not technically a fungicide because it's not killing the fungus. It just sort of disarms the fungus. And it works very well in a laboratory condition. So the trees in the middle are the genetically modified trees. These were transformed using Agrobacterium tumeficans, which has an infectious plasmid. They moved the gene from wheat into chestnut. They selected for it using marker genes, and they grew those up in tissue culture. They got them growing into their greenhouse, and then they challenged them with blight. So the trees on the left are the American chestnuts. The trees on the right were the Chinese controls. And so in this slide, which is the one they picked to show me, by the way, Bill Powell gave me this slide, the Americans were at least as good as the Chinese, better than the Chinese. So it's inherited in the hemizygous state, which means it's on one of the chromosomes, but not on the homologous chromosome, so it's not heterozygous, it's hemizygous. But the prediction is that half of the offspring will carry this if you use the GMO parent as the pollen parent in a cross. So it's a very simple process then to move the OXO gene, the oxalate oxidase gene, from this transformed plant into our breeding populations. In other words, if I could get some of that pollen, put it on my trees here in Tennessee or Georgia, half of the offspring would inherit the oxalate oxidase gene. And you can do the test, it's essentially like a litmus paper. You take a tiny sample from the nut without killing the nut. You take a bone biopsy tool, take a tiny piece of the nut tissue, test it with the enzyme. You can tell which ones have the enzyme and which ones don't. The ones that don't, you can eat them. You don't have to plant them. You don't even have to worry about them. So it works pretty well. It worked well in New York. In Virginia, we did some trials. By the way, it's not deregulated. It's controlled. We can't release it to the public yet. We need permits to use it. We did some trials in Virginia. It didn't work as well in Virginia as it did in New York, probably because it's just hotter in Virginia. The fungus grows more aggressively in high temperatures. So the trees in Virginia didn't respond as well as the trees in New York. So backcross breeding is the part that I mostly do. What time is it?
2 to 7.
I got 2 minutes. No, my pumpkin comes to get me in 2 minutes. So I said that I'm a plant breeder. I said we're plant breeders because I don't do this alone. I have a big group of helpers. The American Chestnut Foundation is primarily a volunteer organization. It's citizen science. We've got thousands of volunteers in chapters from Alabama all the way to Maine. We've got ourselves organized into breeding regions. We've got a New England region, a Middle Atlantic region, a Southern region. This is the plan that we started with 30 years ago, was to start with a Chinese-American hybrid. We call that the F1. Was to make successive backcrosses. Each backcross dilutes the Chinese genome by half. By the time, in this case, arbitrarily, I get to the third backcross, these trees would be 15/16 American. By the way, it doesn't really work like that for a couple of reasons. One, there's 12 chromosomes. The chromosomes break and reform every generation. We select for the American morphotype. So we recover the American genome more quickly than you would predict on this. According to Burnham's plan, Charles Burnham was a plant breeder, a corn breeder. We could fix that by growing these third backcross F2s will vary. This we call the segregating generation, would vary from fully susceptible to fully resistant. And if we could choose just the fully resistant one and cross it to another fully resistant one, all of these would be true breeding blight-resistant trees. In other words, we could fix blight resistance in that B3F3. That's the part that didn't work. In other words, when we got down to this B3F3, what we noticed is that they are still segregating. And the reason for that is because there are many, many more genes than Burnham predicted there would be. So instead of just 2 or 3 genes, there are probably dozens. And worse off, they're scattered on all 12 chromosomes. And so when we look at the genomic map of resistance in these trees, it looks like a patchwork quilt. It's just a patchwork quilt of resistance genes and susceptible genes. But we are making progress. So here are just a few slides of the breeding, the way we do classical plant breeding. The trees are monoecious. They have male and female flowers. We can do the work in the orchard. We harvest the seeds in September, October. We can grow them. We can direct seed them into the orchard, which is actually the best way to do it if you can. You protect them from varmints, but you can plant the seed directly in the orchard. Or we grow them in containers. View of the nursery. We transplant. This is another experiment I'm not going to talk about. Tender loving care, deer protection, weed protection, varmint protection. When they're about 3 inches diameter, which takes about 5 years on a good soil, we try to kill them. So I was going to say we screen them for blight resistance, but what screening is a euphemism for killing them. We give every tree a potentially lethal dose of the fungus. And we do that by removing a plug of bark and replacing it with a plug of mycelium. The susceptible trees develop a canker. There's a beautiful canker. We're trying a method to do this in 1 year in a what we call a small stem assay. It mostly doesn't work. This is the Chinese control. It was supposed to be the resistant tree. It just died, right? Because the fungus is so aggressive. It kills even the resistant tree if the stem is too small. The susceptible tree, this is a tree. This one's dead already. It just doesn't know it. Here's a sibling of that tree that's highly resistant. And by the way, this is Chinese level of resistance. I'm not going to talk about all 3 cankers on there because that was actually an experiment on hypervirulence. But that's a resistant one. That same tree on the right, a few years later, the cankers had healed. And so that tree had a nice long life ahead of it. We've used that one in our breeding program. We've advanced it. And I've got progeny in other orchards of that tree. Parentheses. That individual tree, about a year after I took this picture, died of Phytophthora root rot. No, really, like sad, right? Really sad. And then, well, never mind. The screening process means either they're dead of blight or they don't meet our criteria for selection. In other words, we have a rubric where we look at about 6 or 8 or even 12 different character states for selection. If they're selected against, we cut them down. So the value of the ones that remain is really proportional to the number that we grew. That's why we have to grow hundreds. Eventually, the selected trees, these are selected trees, can be intercrossed. Here we are making intercrosses to produce those F2 generations. The seed orchards are planted at very high density. You can see some older trees in the background, some younger trees that have just been planted. And the reason they're at such high density is because we know we only want the best one out of every 100 or so. So I'm literally planting them 1 or 2 feet apart because I'm going to clobber these things. I don't want to grow mediocre trees. I'm just going to pick the best one. That's really hard to do, by the way, but we're making progress. Some of the trees we have stood the test of time. We're doing long-term phenotyping, which means these trees were screened when they were young. They've been screened again. They've passed all the tests. We're breeding them now. We have progeny testing data for them. In other words, we've grown their seedlings. We've challenged their seedlings with blight. These are the best trees we have. So we've got a program now where we're combining the best trees from Tennessee, the best trees from Georgia, our best by best trials. And last year we had a bumper crop. I predict another bumper crop this year. And so if you want to participate, we're looking for growers. They don't grow everywhere. They don't like clay. They don't like floods. They don't like Phytophthora. What? Right. But if you've got them on a good site, they're very, very fast growing. They'll grow as fast as a tulip poplar or a white pine. Really, they can keep pace with those trees. So I'm over time, but I'm certainly willing to answer questions. Or not. Ask me.
Earlier in the presentation, when you talked about the original blight, what was the time span in which the original American chestnut?
So the question was, what was the time span? You know, the blight was first detected in 1904 in New York City. Obviously, the introduction was prior to 1904. We know now that it came from Japan and was probably on nursery stock from Japan, people moving plants. That's how plant diseases move. The Japanese chestnuts make a great big nut. American chestnuts are small, and so they were popular in the mail order trade. You want to plant chestnuts to eat, you plant something that makes a great big nut because you got to peel them to eat them, right? And so many, you know, there's this idea that it spread sort of like oil spreading out over water across the landscape. Not really true because they were mailing it. So by 1904, there were already foci in the Carolinas and Virginia. Then it coalesced into this pandemic. It came through Chattanooga in the 1930s, and by 1950, more or less, it had run its course. Similarly, in Italy, it started in the 1930s, a little bit later, and by the 1970s, hypovirulence was starting to kick in. And so a similar pandemic, different trajectories though. So in Europe, the trees were pretty much cured by hypovirulence. In North America, not so. There are some bizarre exceptions for that. In northern Michigan, not the northern peninsula, but in the northern part of the southern peninsula, there are some recovering populations of American chestnuts. Now, it may be that those populations were introduced. In other words, they're not truly native there. Some pioneers, took them up there and planted them. But hypovirulence is effectively controlling chestnut blight in some of these places. So to answer your question, it was about 50 years. But by 50 years, it was gone. I had a student a few years ago who did an oral history project where she interviewed eyewitnesses. And she looked at the map and she said, well, if there's any eyewitnesses alive, they're going to be in Georgia and Alabama, North Carolina and Tennessee. And so she was able to interview about 20, 25 eyewitnesses to the chestnut blight. It was very interesting. It was heartbreaking, but very interesting. We heard some of the same stories over and over again. We heard about hogs, that they would just earmark the hogs and then they would turn them loose in the woods. And when the chestnuts were ripe, the hogs were so fat, they were just lying down up there under them chestnut trees. We just loaded them on the cart and drove them home. Yeah. My feet were so tough, it was the Depression, right? I had feet so tough I could stomp a chestnut burr. Heard that a bunch of times. Another question for me.
Are somewhat blight resistant or the most blight resistant trees being planted in national forests, national parks in many areas? And are there areas there where you don't have a problem with the root rot?
So that's a great question. So it was, are we cooperating with the national forests and the state forest? The state forest is patchy. That depends on the state. And yes, the National Forest, absolutely. We are collaborating. We've had a memorandum of agreement with the National Forest for about 20 years. We don't get any money directly from them, but we do collaborate. And that's in part how we know that the B3F3s are not fully blight resistant, because the Forest Service does a very good job of tracking and measuring their trees. So they have planted thousands of our hybrids in national forest lands, in forest plots, where they measure incidence of blight. And so we've done progeny testing from our breeding program in the national forest. Very important point, by the way, is that from plant breeding, we're doing artificial selection. The idea is that we have to grow these in a natural environment in a way that natural selection will take over. If we're going to be successful at this, and I'm talking about long-term success, these trees have to be able to reproduce on their own in the wild, whatever that means. And so that last round of selection really has to be in the environment where you plan to grow them. So Georgia, very different than Pennsylvania. Pennsylvania, very different than New England, right? I mean, from Pennsylvania to Connecticut, very different environment. So we're testing these trees across a broad range of environments. But yeah, the Smoky Mountains is a national park. Absolutely no work in the national park. I mean, you're not even allowed to. I shouldn't exaggerate. You can touch them, but you can't pick them. You can't collect a leaf without a permit.
You can't plant the leaf.
No, absolutely not. And so we can rationalize that by saying the Great Smoky Mountains National Park is going to be our reservoir of susceptibility. And there's a rationale for that. If you know about plant disease and disease breeding, there's a rationale for leaving a part of your population susceptible, and that has to do with the evolution of the pathogen itself. So if we might be better off in some eventual future having not planted any American chestnuts in the national park. But no, we don't have permits to plant in the park. By the way, we have got permits to collect scion from the park. And I'm supposed to send them a report every year, forever. That's on Facebook now. The Park Service employee is going to send me a note. Where's your report? Right. Did I answer that question? Another question for me. Yes.
I see saplings regularly enough when hiking the mountains. Is there any characteristic besides the size that might make one stand out and be worthy of?
So the question was, he sees saplings when he's hiking. Like I said, the chestnut blight kills the top but not the roots. And so they've been able to resprout from the base. Occasionally, there are escapes, a tree that escapes blight long enough to bloom. And we do occasionally see seedlings. Even though I said before there's been no reproduction, no natural reproduction. There's no evidence of recruitment into the population. Not really true. Occasionally we see a sapling or a seedling. And we know that, for instance, if they come up in the middle of a Forest Service road. Right? There's a sapling, it's in the middle of the road, right? So we do see them occasionally. What makes them really special is if you see a tree that has blight and has had blight for many years but didn't die. That's a large survivor. And it varies from state to state. There are some places geographically, there are some places in Virginia, for instance, in sort of the Piedmont of Virginia where they really ought not to be, where there's a disproportionate number of these large survivors. In parts of Maryland, eastern Maryland, there are some clusters of these large survivors, and there are some different hypotheses about that. One is that it's just hypovirulence. Two is maybe they're not really American chestnuts. We know, for instance, I'll just give you an example. Thomas Jefferson was planting European chestnuts at Monticello in 1780. And they've been growing, the DuPonts in Delaware had whole plantations of chestnuts. There was a chestnut breeder in eastern Pennsylvania, right near Philadelphia. Colonel Sober had Sober's chestnut farm where he was actually breeding American chestnuts and European chestnuts for the nut quality back in like the 1890s. He ran a train from downtown Philadelphia to his farm in the fall with a dining car so that you could go chestnutting. So you have dinner on the train or lunch or whatever it was, brunch. You drive the train out from Philadelphia onto his farm. He had a lake with swan boats and the chestnuts were on the hill over there. You could pick the chestnuts, take them back to Philadelphia. Very, very fancy. Chestnut blight ended all of that. But the point is that these populations of chestnuts in Virginia, Northern Virginia, New Jersey, Pennsylvania, they might not be all American chestnuts. We don't know. But the ones you see hiking around here are surely American chestnuts or chinkapins. Come look at the chinkapin before you go and learn to tell a chinkapin. Another question for me. Yes?
I'm a little confused about the leaf rot now. It's indigenous, right?
It probably also comes from Asia, in the sense that it's not a North American origin. It was introduced. One of the weird things is that we blame them, they blame us. It's like we don't really know where it's from. One of the ideas is that most of the plants that exhibit strong genetic resistance are East Asian. So we think it's probably of East Asian origin because of this hypothesis that through coevolution, those plants have evolved some sort of resistance to it.
But it's not everywhere. It's in pockets. Is that the reason why?
I don't know. And so here's the risk, is that way up in the mountains in the national forest, it's probably not. Until some idiot like me takes a plant from the nursery up into the national forest in the mountains and plants it. Now it's there, right? And so it's we humans moving plants that move plant diseases. I'll say that again. Humans are the main vector of plant disease transport around the world. We move our plants, we move plant diseases with them. And so, it was a very wonderful, brilliant idea to grow the trees in the nursery. We can grow 1 million of them. Let's do a big forestry planting. We know now that we've introduced Phytophthora root rot disease into some of the national forests, not just on the chestnut trees. What about all those loblolly pines? Right? Oh yeah, and so the loblolly plantations, we know now pretty much for sure if it's a loblolly pine plantation, American chestnut just won't grow there because they're all infected. They came from the nursery, yeah. So, but very good question. The question was, is Phytophthora anywhere? The last really significant mapping that I saw was back in like the 1940s. And then I think people just sort of threw up their hands. Although a few years ago it was recorded in the Pittsburgh, Pennsylvania area. Apparently Phytophthora cinnamomi had not been recorded in Pennsylvania until about 5 years ago. So maybe it's moving north. It's limited by cold. Among the crops that are very, very susceptible, Fraser fir. The Fraser fir nursery industry has had to completely retool. Until about 10, 15 years ago, most Fraser fir nurseries grew the seeds in the ground, and then they would dig them and plant them in the... Fraser fir is popular for Christmas trees, right? Now, 100% of the Fraser fir nursery industry is in containers off the ground. They grow them in a soilless medium, in a pot, and the pots are on benches 3 feet off the ground so that they don't get splashed. And so they're growing a Phytophthora-free seedling. They had to do that. The Fraser fir nursery industry completely transformed because of Phytophthora. Another question for me. So if you want to know about chestnuts as a nut crop, invite me back in the fall, like late October, early November, and we can talk to the chef across the street here, and we'll do a chestnut dinner. We can do a chestnut dinner. Ask me.
That's one last question.
Sure.
It seems like years ago, I remember reading about a small subgroup of chestnut preservationists, you might call them, that wanted to go with only 100% American.
Sure.
Using and not using any of the
hybrids, right? And so they're basing their work on those large survivors.
To be productive at all, or they still doing that.
Yeah. No, and so we cooperate. Yeah. They don't like the hybrids because they're exotic, and there's a good rationale for that. But we know that not all American chestnuts are uniformly susceptible.
I mean, I think I would read that maybe some in Michigan.